Welcome back to DC Tutoring. I'm Drake, and let's just jump right into it. This is going to be Chapter 2, Amino Acids, the Structures and Biomechanical Properties. So our objectives for this chunk of material is going to be describe the structure of amino acids at physiological pH. If you can remember, what was our physiological pH? Normal for our body to survive, that was 7.35 to 7.45, with special reference to their R groups and their properties in amino acids and proteins. Remember, we also talked about last time those R groups, where we'll have, for example, methyl. We'll have our structure here at the end, and then we'll have it connected to an R group, so we'll be looking at those R groups and their properties in amino acids and proteins. I also want to describe the effects of pH changes on if I can say the word, ionizable, on groups of amino acids that you can create and turn into ions. We're going to classify the 20 amino acids found in proteins with reference to their physical properties, such as if they're hydrophobic, hydrophilic, or their ionization, etc. Describe the structure, function, characteristics of the amino acids, and describe modifications of amino acids in proteins. Okay. So the general structure of standard alpha amino acids at pH of 7. Remember, in our body, we're looking at that neutral pH. So, proteins are composed of amino acids. In humans, the L isomers of 20 amino acids construct proteins. Break these down a little bit. So when we talk about L isomers, right? An isomer is referring to same chemical makeup, just different shape. So if we look at some So if we look at isomers, we have molecules that are the same. So C2H6O, and that can be arranged as ethanol or dimethyl ether. So it's just saying that we have the same chemical formula, but different possible structures. So in humans, we're going to be using the L isomers. That's what we use in our bodies to construct our proteins. The side chain is designated as R group, which is attached with the alpha carbon atom. Alpha carbon atom, make sure we get a good definition for what that means. So the alpha carbon refers to the first carbon that attaches to a functional group. So the first carbon that attaches to a functional group. We talked a little bit about those functional groups earlier. The functional group is going to be what is going to be actually doing the reaction there at the end. Okay. The other three bonds of the tetravalent alpha carbon atom are covalently bonded to an amino group. So if you remember covalent, we talk about covalent bonds. Think back of what that means. It means we're sharing those electrons. Bound to an amino group, a carboxylic acid group, and a hydrogen atom. So the alpha carbon atom. So we'll also look at the amino groups, carboxylic acid group, and the hydrogen atom. We'll see kind of the structure of all these amino acids. Thus, the alpha carbon atom of a typical amino acid contains a chiral carbon center. Chiral carbon center. Chiral, it's a little confusing when you talk about these things. So the rule of thumb is this. It's a really good definition. So the chiral carbon centers are carbon atoms that are attached to 4 different things. Now that's where we'll end it as for the purpose of this class. So, for example, this is what we really want. Carbon centers. Our carbon atoms are attached to 4 different, we'll say substances, because that's a little bit easier definition. So that means we have a carbon, and so let's say we have our amino group, we have our carboxylic acid group, we have our hydrogen, and we have our R group, right? So it's connected to 4 different things, chiral carbon center. And these are what we'll see within our amino acids. Is because it contains all 4 different groups attached to it. However, exceptions exist. Exceptions exist. The amino acid glycine contains 2 hydrogen atoms and does not have a chiral center. So in some cases, so you'll most likely get asked the question at some point right here. Beautiful test question. Which of the following amino acids does not contain a chiral center? What is our answer? Glycine. Perfect. That'll pop up somewhere, I promise. The side chain, the R groups, plays a major role in amino acid biochemistry. Some R groups have neutral charge, and some of them have either positive or negative polarities. Remember, when we talk about polarities, we're talking about our different interactions when you have kind of like ions. The positive and the negatives will attract, and they can create reactions. This imparts a role in the solubility or ionization of the amino acids in biological fluid environment. Biological fluid environment, remember, our entire body surrounded by that wonderful H2O, and are often altered during many biochemical and or physiological changes in our body systems through ionization, or during alterations in pH environment, and thus influence their biochemical and physiological characteristics, reaction capabilities, physical properties, all of that stuff. Basically, all this is saying is that while they do have like a typical state where they are going to be positive or negative or neutral, depending on the environment that they're in, they can be changed. They can have some changes made into them, so that way they do different, play different roles or react differently with the things around them. Okay. Here is our beautiful alpha carbon. Remember, that is our chiral center, chiral carbon center. And what was the one that didn't have the chiral carbon center? Our glycine. Keep that in mind. So our carboxyl group, COO, our primary amino group, H3N+, our R group, which will be later on, and then our H. Okay. So chirality, asymmetric carbon, and this is what's really nice about making sure that you go and you find those definitions. It might be a little tedious, and you're like, oh, it might be listed later on. Now that we've already listed it here, we're going to get this beautiful repetition where it's an alpha carbon is bonded to 4 different groups. Okay. Now we have stereoisomerism. This is kind of like the mirror images of stuff. So if this was a mirror right here between the two, see how this OH kind of flops over, and you see that mirror image of it where these two flip just like that. It's a mirror image of us. So the D isomers exist out there, but for pretty much all of these, we're going to be focusing on our L isomers. So if you get questions, you can have questions like, which of the following is going to form this reaction in the human body? And he'll throw in a couple of those D isomers in there, And in pretty much every single case, depending on the wording of the question, if it's in the human body, we're going to ignore those D isomers because we know that we want to focus on our L isomers. Okay, amino acid classification. So different chemical properties of amino acids are due to the presence of different side chains, or those R groups. So if we look at the structure, all of this up here is the same for every single one of our amino acids. What makes them different and unique and gives them their properties is that R group. Okay? Some of the side chains are polar, nonpolar, aromatic, positively charged, and negatively charged. And we'll go and we'll look through all of these. So amino acids ionize in aqueous solution. When we talk about aqueous solution means that it's water. Ionize, make sure we get a nice definition for ionize. It's just converting any of it into an ion. So remember those two that we talked about. We have cations and anions. Cations, I always remember that cats, I like cats, cats are positive. Anions, that A suffix is usually no or bad, so the anions are negative. So what the carboxyl and the amino groups can ionize. So the carboxyl and amino groups, remember, are our carboxyl and our amino group. These guys can ionize. Amino acids can act as weak acids and weak bases. Remember when we talked about acids and bases, what were we referring to? Acids are, well, he has it written right here. Acids are our proton donors, and the bases are the proton acceptors. Remember when we talked about weak versus strong? Weak versus strong. Weak, it's talking about how well it dissolves, right? So a weak one doesn't dissolve fully, and a strong almost entirely fully dissolves. And then, of course, we also talked about when you put a weak acid into it and it dissolves, that weak acid, put that into solution, and you get that hydrogen plus its conjugate base, and vice versa. So a weak base, you get the OH plus its conjugate acid. Good review. Okay. So the R groups of some of the amino acids can be ionized. So we're looking at, it can be a carboxyl or an amino group, or some of the R groups all can be ionized. So all of these ionizable groups affect the net charge on each amino acid. So here we're going to look at some of our amino acids broken up. So our non-polar R groups here. And remember glycine, what was special about glycine? Non-chiral carbon center. Okay, because we have the two hydrogens. So hydrogen, that's what makes it non-chiral. Okay, so our non-polar ones is these hydrogens. And again, you don't necessarily have to memorize these structures, especially since if you look at these, on every single one, all of this is exactly the same. If you really want to look at something, if you need something to really study and to differentiate them, look just at these bases here, because everything else is going to be the same. All right? So we have glycine, alanine, proline, valine, leucine, isoleucine, and methionine are all of our non-polar amino acids. And let me actually see if I can find something real quick for a little helper to remember all of these. While it's pulling up, we'll continue. I'll come back to it if I can find it. Here we go. There's some little word play on word things that you can use to help remember some of these. I don't think there's one specific for nonpolar, but yeah, we'll get into all of that later. Okay. I'd recommend writing these out a couple of These also all come with 3-letter abbreviations. Remember, proline is pro, methionine is met, isoleucine, I believe, is IL something. I don't quite remember all of the 3 letters, but you'll start to see those pop up later on. It's good to kind of make a nice little chart with all of those in there. So important notes about some of the amino acid structures. Again, glycine is the simplest amino acid. It does not have the chiral center. All other amino acids are asymmetric. Asymmetric. What does that mean? It means no matter how you split them, they're going to be different. You're not going to see a repeat on the other side. One hydrogen atom of glycine is replaced by CH3 to make alanine. Valine, leucine, and isoleucine are branched chain amino acids. Some wordplay for this. You can live on a branch. That's helps us to remember our branch chain amino acids, LIV, valine, leucine, and isoleucine. Amino acids containing aromatic rings. These are our ring structures as their side chains. This one, to help remember, it's your pal Tyrone will trip you in the ring. Remember our aromatic rings. These might seem kind of silly, but honestly, this is how they're going to teach you for boards as well, to remember these. And if you can remember these, this is all free questions, free points on all of those tests. Live on a branch. Your pal Tyrone will trip you in the ring. Our polar uncharged. I feel like he doesn't. I'm trying to remember if he asked specifically about the polar ones. The way that I remember doing this, a lot of this was kind of looking at trying to make up little similar to these, make up little word plays to help you remember these. If you come up with one yourself, it's going to be a lot more memorable, and it's going to help you really ingrain it into your brain and practice it. So I'd highly recommend looking at these. See if you can come up with something, merge the letters all together. You can even throw it into one of those. I know there's a Scrabble word finder, whatever, on Google. Throw the letters, the first letter of each one in, and see if you can come up with a word that uses all of the letters. Fun stuff. So important notes about amino acid structures. Serine, threonine, and tyrosine contain a hydroxyl group as their side chain. Cysteine and methionine are sulfur-containing amino acids. These sulfur ones say to be in MC, you have to have sol as in sulfur. Asparagine and glutamine contains amide groups. And the sulfhydryl groups of 2 cysteines can form a disulfide linkage. This will be a big question that they love to ask. That the sulfur-containing ions, especially like cysteine and methionine, can have a disulfide linkage. That's a good question that comes up just almost all the time. Which of the following amino acids can form a disulfide linkage? Answer: cysteine or methionine. Either of those would be acceptable answers. More commonly cysteine. Asparagine and glutamate, which both of these actually also have. I'll get into that later. Where both of these do have more acidic forms, but glutamine has glutamic acid. But we'll look at those a little bit later. Positively charged R groups, lysine, arginine, and histidine. And these amino acids, lysine, arginine, and histidine, are also basic amino acids containing amino group as their side chains. So you can see those amino groups within their side chain all through here. Negatively charged R groups, aspartate and glutamate. These are our acidic amino acids. Good way to remember the acidic is acidity of as and glutes. Some of these have the word play, others you might just need to go through and kind of make up your own. These are the ones that I learned that I used, and they're definitely helpful in helping to remember some of the key ones. Okay, oxidation of two molecules of cysteine form disulfide linkage. We'll get more into these reactions, but oxidation is an important reaction to remember. You're gonna see a lot of this, of oxidation and reduction. In your undergrad, you might have seen this used oil rig, where oxidation is loss of electrons and reduction is gain of electrons. You'll get a lot of practice with this stuff as we keep going on. So see how when we get the disulfide bond, we lose those hydrogens, we lose those electrons, and we form that disulfide bond. So the two cysteine, pay attention to this, cysteine becomes cysteine. There's lots of little word changes that can bite you in the butt when you're taking exams if you don't recognize those. So pay attention to those little differences. So the disulfide bonds between the cysteines, the cysteine residues, stabilize the structures of many proteins. That's why it's so important. These disulfide bonds are really important for stability of a lot of our proteins. For example, insulin. As we know, insulin in the body, you'll learn a lot about insulin in your physiology courses in tries 2 and 3. But you can see how all of these are bound together to form our insulin. And but we use the disulfide bonds in insulin for its stability and its structure. So you can see all through here, disulfide bond, disulfide bond, disulfide bond, to maintain the proper structure of our insulin. We'll get into and mention this later, but when you have a chain of amino acids like this, we call it a polypeptide. Polypeptide chain. We have our amino acids, which are our peptides. They will join together to create the polypeptide chain, and then the polypeptide chain will get folded or joined with other things to form our proteins. And we'll get into that when we talk about the formation of proteins. We'll talk a lot about that. The Zwitterion. This is one. Again, I want a nice definition for the Zwitterion. So let's jump to our wonderful Google. So Zwitterion is a molecular ion having separate positively and negatively charged groups. So the Zwitterion can act as either the proton donor, the acid, or the base, a proton acceptor. I believe, if I'm not mistaken, he talks about the Zwitterion in his lectures. I know I saw it here earlier before when I was reviewing. There it is. Yeah. So it's a amphoteric, which is both acidic and basic, and he gives a couple of examples in here of its ionic form, where, so see in this case, it bumped off that H, the hydrogen, and gave it back to the amine group. So now we have, again, it can either accept or it can drop off the hydrogen ion. Yeah, and here's the same thing. So a little bit more on the Zwitterion. The amino acids are often characterized by the pH at which the net charge is 0. This pH is called isoelectric pH, or the isoelectric point, PI. You'll see on a lot of the science courses, there's just a lot of AKAs for stuff. Knowing the AKAs is really, really important when understanding questions. So this pH, the isoelectric pH, or the isoelectric point, is when the net charge is 0. Titration of glycine. Let me see how much we got left. We got a little bit left, okay. So in chemistry, acid-base titration is used to determine the concentration or strength of an unknown solution. So we have an unknown solution. We're thinking, okay, is it acidic or is it basic? Let's find out. By neutralizing the acidity or basicity using an acidic or basic solution of known concentration or strength. One simple example of titration is described here to give a basic idea using the simplest amino acid, glycine. However, one can use other amino acids to observe its changes in ionic properties during acid-base titration. This is what we talked about, how if we change the pH, certain our amino acids are going to change slightly, the structure is going to change slightly, which will then impact how they respond and how they react with the things around them. In the present case, glycine is titrated by adding known concentrations of a base or hydroxyl equivalent. Glycine contains two ionizable, there we go, contains two ionizable, it's a little late, I'm sorry, functional groups. One amino acid group and the other is a carboxylic acid group. So our amino group and our carboxylic acid group. At a very low pH or in strong acidic environment, remember low pH means that we have a lot of hydrogen, it's a strong acid. Glycine exists as a fully protonated form, so it's loaded up with the protons. Our OH concentration is at 0. As the titration is started by addition of alkali or base, so an alkaline solution is the same thing as a basic solution, saying the exact same thing. The carboxylic acid group loses the protons because the hydroxyl ion combines with the proton of the carboxylic group and produce water, leaving the Zwitterionic form of glycine. At the midpoint of this stage, when the OH equivalent is 0.5, the concentration of the proton donor form of the Zwitterionic form of glycine exists in equimolar concentration. So moles is one of the ways that we measure concentration. So moles, it's the SI base unit for the amount of substance. So it's about if you have a certain number of moles within a solution. So equimolar concentration, meaning that, is it just saying that the concentration of everything is at equilibrium, or an equilibrium between these two forms are reached. Now, as a titration is continued by addition of base, another point is reached at pH 5.97. or the OH equivalent of 1. So all of continued by addition of base, another point is reached, the OH equivalent of 1. So meaning that all of the OH is dominant at this point. So at this point, the proton is now completely removed from the carboxylic acid group, followed by the beginning of the removal of the second proton from the amino group of glycine. As the titration is continued further, a second point, pK2 9.6, is reached. In this stage, the Zwitterion form, and the H2NCH2COO form is present in equimolar concentration. Further addition of hydroxyl equivalent finally completes this change and produces 100% of this form at the OH equivalent 2. Isoelectric pH is calculated by adding the pK1 and pK2 values divided by 2. So why knowing this kind of titration of amino acids are important? So there's a lot of words. You don't focus on trying to memorize all of this. This is a lot of complicated mumbo jumbo. So let's really focus. What we want to focus on more is like the why. Why is this important? Okay, not necessarily this whole process, but this is going to give us an idea about the net charge of the molecule at various ionic environments, which has implications on its biochemical reaction characteristics at various ionic conditions that may occur at different situations arising from normal reactions. Three-dimensional spatial orientation of the molecule to be interacted with another, or to the development of certain diseases. So what we know then is that when we're looking at this, we can see what percentage, in what form this ion is, whether is it positively charged, is it negatively charged, and at what different pH levels, essentially, is it at that form. Because when we're not in equilibrium, so if we're sick, our body tries to maintain homeostasis, meaning that we're all completely level, that we have a balance in all things. If we're out of homeostasis, then our pH levels are going to be slightly skewed, which means, especially like it can be skewed in specific areas of the body. So we're going to have different pH levels in different parts of the body. Then we can have different interactions due to whatever has taken us out of homeostasis. And this is our beautiful curve. This is the curve, the titration curve, that it was telling us, that was talked about in that entire chapter, that entire massive paragraph. So you can take a look at this. Again, this is something, titration is something that, and calculating titration is a little bit complex and really requires a little bit more time dedicated to it. If I'm not mistaken, Ninja Nerd, I believe Ninja Nerd, yeah, he has some great videos on general chemistry and titration. So the weak acid and strong base and the strong acid and base titration. I would highly recommend coming to watch these videos, as he really walks through the process. If you have the time to go through these, it'll really help deepen your understanding. And it might not be like the most important thing, but understanding kind of what each of these individual things mean, what the PK1 versus the PI, the PK2, what those mean, could help somebody really understand this a lot better and be able to answer the questions if he asks whatever questions the teacher might ask about those. So the amino acids are polymerized by condensation to form a peptide bond. Here's another, earlier we saw oxidation reduction. Now we're going to have condensation reactions. Condensation, we're having water. Let me triple check that, because there's condensation reactions and hydration reactions. I want to make sure. Each molecule is combined to form a single molecule. A small molecule, often water, is usually removed during condensation. Yeah. Always double check your work. So the condensation reaction, we're going to have two molecules coming together, and water comes out when a bond is formed to form that peptide bond. Remember, I talked a little bit about this earlier, where the amino acids will join together and form peptides, and they'll form peptide bonds to form a polypeptide chain. And that's what we're talking here. Interactions in proteins. Externally available functional groups of molecules, from proteins to carbohydrates to lipids, can interact with the amino acid side chains and produce hydrogen bonds. When we talked about the hydrogen bonds, remember, most commonly it was with oxygen, but any of those hydrogen molecules that are hanging around can form hydrogen bonds. So we have here, as we form our polypeptide chain, each of these forming these different bonds here. Each of these are our R groups. So we have our single H right here. This was our glycine. CH3 was our alanine. Yep, it was our alanine. We had the OH, I believe that's our tyrosine that we have there as well. Yep, there's our tyrosine. So each of these, as they continue to combine to each other, you have the amino terminal end and the carboxyl terminal end, and we'll be adding here on that carboxyl terminal end and growing this side chain, this polypeptide chain, for as long as we can. And we'll really get into how this whole process happens later on, when we look at the DNA polymerase, how it creates RNA, and then the RNA creates mRNA, and then the tRNA will bring the amino acids. When we look at the transcription and translation process of our proteins. So modification of amino acid R group structures regulate biochemical functions, form signaling molecules, helps in targeting or anchoring to membrane, and enhance protein-protein interactions. So covalent modification of amino acid structures. These are all of our modifications that can happen to our protein. So hydroxylation, carboxylation, methylation, acetylation, phosphorylation, addition to carbohydrate moieties, in addition to lipid moieties. This is one. Get good definitions for every single one of these. Hydroxylation, adding of hydroxyl groups, is an oxidation reaction in which carbon-hydrogen bond oxidizes into carbon hydroxyl. So we're adding that oxygen right there. These, if you can understand these really, really well and know what each of these are, chemical reaction with carboxylic acid is produced by treating substances with carbon dioxide. So this is adding a carbon dioxide. Methylation, this is adding a methyl group. Acetylation, introduces the acetyl functional group, which is the CH3CO. If you can understand all of these different groups, as you start to get into later on down the road, we really start to look at the enzymes that do all of these different reactions. And each of these, like the methyl, phosphorylation, acetylation, carboxylation, hydroxylation, these will come up in the names of the enzymes. So if you know what it is that's happening, it's so much easier to understand what the enzymes are and what it is that the enzymes do and what the next steps are going to be in those reactions. So highly recommend to spend some time, make flashcards of these, and really, really know these. Know the definition well, know what it is that's being added or taken away, so that way later on down the road, the enzymes are so much easier for you to learn and to remember. Okay, so covalent modification of various proteins are important for the regulation of their biochemical and physiologic functions. Understanding these concepts of regulation also established the basis of development of pharmaceutical therapeutics. Here's our modifications. Adding the carbohydrate moieties, adding the lipid moieties. I'm not going to spend a whole lot of time on that. I'd come back and read this. Just go through it and do what we've been doing. Hydroxyproline, continuing down. Modification of the amino acid. This is the phosphorylation, acylation, methylation, N-methylation, methylation, and adenylation. So again, what is being added to each of these different structures? So the phosphoserine, adding of the phosphate group, adding the CH3, that methylation. And that's it for this chunk of material. A lot of this looks really, really complicated, and it can look really gross, just because of like all of this stuff that's there. Don't get too caught up in seeing all of these and shutting down and not knowing what's going on. Focus right here. What's up here? And understand these words, what they mean. Where phosphorylation is going to add a phosphate group. The acetylation is going to add that acetyl group. The methylation, and methylation is going to be adding it to a different part of the molecule, or the methylation will be adding the methyl group. Adenylation should be adding, I don't remember what exactly group that is. I think it's adding it's having to do with with ATP. Adenosine modifies molecules covalently attached to the amino acid, yeah. So that's adding the adenine group, which you'll learn more about. It's part of ATP and that whole system. Just focus on these words specifically, so that way you can understand the definitions of these. And when you start to get asked questions about it, especially later on with the enzymes, like I was saying, you'll understand what the function of those enzymes are, and a lot of this will start to click into place as we continue to go through everything. Thanks for watching. Remember, study hard and good luck.